Good morning, everyone. How are you today?
Good. How are you?
Okay, thank you. All right, folks. Well, we'll get started here. I do expect attendance to be pretty light, but I thank you for coming out this morning. And I do hope that your Thanksgiving break is an enjoyable time for you and your families. So we're going to continue in chapter 12 this morning. If today goes anything like my other class that I teach, we'll probably end up not quite finishing this chapter, but that's okay. We can make up for that after Thanksgiving. So let's get started here and see how far we can get. Okay, so you may recall from last time that we were talking about these electrochemical gradients. Okay, and I gave you some examples and tried to show you how example one was kind of like a taller hill or a steeper hill than example two. Okay, so I'm not going to rehash that, but certainly if you have questions, feel free to ask. So what I am going to do, the topic of electrochemical gradient was a subtopic under ion channels. And so we need to talk a little bit deeper about ion channels, okay? And so I'm actually going to begin our conversation this morning. I'm going to begin our conversation this morning talking about figure 12.11 in your textbook, figure 12.11. Okay, so obviously as you look at figure 12.11, this figure is trying to introduce you to the various types of ion channels that exist and which can permit ions to move back and forth across the plasma membrane of cells. So what I wanted to do is document these ion channels in your notes and just ask you to put your eyeball on the image of the channel on figure 12.11 as we document these things. The first type of ion channel is a leak channel. Okay, leak channels are made of proteins that randomly open and close, In other words, for leak channels, there is no obvious stimulus that causes the channel to open or close. There's no obvious stimulus for that. These channels simply open and close in unpredictable ways, okay? These are made of proteins that open and close randomly, okay? And so with that, there's some additional information you have to be aware of. It turns out that Mother Nature has designed it so that there's two major types of leak channels that are important for understanding how the nervous system functions. That would be sodium ion leak channels and potassium ion leak channels. Well, Mother Nature has engineered cells of the nervous system to have more potassium ion leak channels on average than sodium ion leak channels. Okay, the plasma membrane of neurons has more potassium ion leak channels than sodium ion leak channels. And as we will get into a little bit deeper into our conversation today, the appreciation of this fact that there are more potassium ion leak channels than sodium ion leak channels goes a long way to helping you understand why the resting membrane potential of a neuron is what it is. Okay, so hold on to that thought. So not only do neurons have more potassium ion leak channels than sodium ion leak channels, it is also simultaneously true that if you compared a potassium ion leak channel to a sodium ion leak channel, so you had one potassium ion leak channel and one sodium ion leak channel, the potassium ion leak channel is leakier than the sodium ion leak channel. Okay, so potassium ion leak channels are leakier than sodium ion leak channels. So what I'm saying to you is that if you had a potassium ion leak channel and a sodium ion leak channel open for the same period of time, let's say 60 seconds, it might be the case that 1,000 potassium ions get through its leak channel, but only 250 sodium ions get through its leak channel. So the potassium ion leak channel is leakier than the equivalent sodium ion leak channel. Okay. So I'm going to put into your notes, appreciate these statements to help understand resting membrane potential. Okay, and I am going to be explaining resting membrane potential later today, so I'm just kind of foreshadowing the pieces of information you have to have in your mind to understand what the resting membrane potential is all about. Okay, that's the first type of ion channel of figure 1211. The next kind of ion channel is a ligand-gated channel. Okay, ligand-gated channels are made of proteins that open or close in response to the binding of a specific molecule to the channel. Okay, these are proteins which open or close when a specific molecule binds or detaches from the channel. When the molecule binds, the channel opens. When the molecule detaches from the channel closes. Okay. And so I want to give you an example here. And before I give you the example, I'm going to ask you folks a question in the chat window. Pardon me. So here it comes. Here's my question. What was the name of the ligand-gated channel that we discussed in chapter 10? Chapter 10 was the muscle chapter. What was the name of the specific ligand-gated channel that we talked about? What do we say it was called? Excuse me. Does anybody remember that? Okay, let me give you a hint. Let me give you a hint. My hint is this. The name of the ligand-gated channel was talked about when we were talking about the neuromuscular junction. Okay, does that help at all? Okay, let me give you another hint. All right, okay, here's another hint. The name of this ligand-gated channel, the name starts with the letter A. Starts with the letter A. Any takers out there? I know it's one of those things where you got past the exam, so all the knowledge just floats out of your brain, right? Okay, what is the name of the molecule that attached to a receptor, hint, 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 in the membrane of the muscle cell to cause the muscle cell to generate its own electrical signal? Okay, all right, looks like we're maybe a little tired this morning, so let me help you along the way. Do you remember the conversation in the neuromuscular junction where the neuron releases acetylcholine. Acetylcholine floats across the synaptic cleft and attaches to, drum roll, the acetylcholine receptor. The acetylcholine receptor is an example of a ligand-gated channel. Okay. So folks, let me just ask you quickly here in the chat window. Do you recall the acetylcholine receptor that we talked about, a couple weeks ago? Okay. All right. Well, let's continue here. So, what we're trying to do is connect the dots, right? We're trying to take ideas that we've seen before and connect it to new topics in chapter 12, right? Because if anything, your education is about trying to connect the dots between massive volumes of information, okay? So the acetylcholine receptor present at the neuromuscular junction would be one example of a ligand-gated receptor. Okay, there are many ligand-gated receptors throughout your body. They will bind things like neurotransmitters. So the acetylcholine receptor, is a receptor that binds neurotransmitter. You can also have other receptors and other target organs of your body that bind a variety of hormones that support endocrine function. Okay, and you'll get much more of that in AMP2. Okay, third type of channel in figure 1211 are the mechanically gated channels. Mechanically gated channels are proteins that open or close in response to some kind of physical stimulus. That physical stimulus is typically touch. So when someone touches your arm, you're going to feel that because of the opening of mechanically gated channels embedded in the plasma membrane of neurons that are stuck in your dermal layer of the skin. So touch, vibration. If you've ever mowed the yard and you had your hands on the back handle of the lawnmower as you walk, typically because you're moving and the lawnmower engine is moving around, there's a gentle vibration to the machine that you can feel. Well, you're going to feel that because of mechanically gated channels. Tissue stretching or compressing in some way. For example, if you were to pinch your skin a little bit, let's say you won the lottery and people say, oh, I won the lottery, pinch me, it can't be real, right? Pinch your skin a little bit. If you compress the skin or stretch the skin a little bit from its natural shape, then you will open mechanically gated channels and your nervous system will be aware of what's happening at the surface of your arm. So these kinds of channels are made of proteins that open or close in response to a physical stimulus, such as touch, vibration, pressure, and tissue stretching. Okay, so, it's probably the case this weekend, particularly with the holiday coming up, that you might see some old friends or family members you haven't seen in a while. So you're probably likely to get a hug or a kiss. You're going to be aware of the sensation of the hug or the kiss because it's all going to start with mechanically gated channels opening in response to that hug, which is going to allow ions into the neurons that are embedded in the dermal layer of your skin. Those neurons are going to generate graded potentials, graded electrical signals, which will eventually trigger an action potential in the axon hillock of the neuron, which will spread up your spinal cord, up your brainstem, into your higher order brain structures, and all of a sudden you'll be like, oh, Aunt Nancy's giving me a hug, okay? All right, next, the fourth and final one are the voltage-gated channels. A voltage-gated channel, these are all proteins. This is made of proteins that open or close in response to a change in membrane potential. Remember, membrane potential is the difference in electrical charge on the inner and outer surfaces of a membrane, okay? So let me ask you in the chat window, here we go. What specific voltage-gated channel did we talk about in chapter 10? Okay. Think about that neuromuscular junction. What did we talk about in chapter 10 that was an example of a voltage-gated channel? What was the name of that? We had sometimes abbreviated it VGCC. What was that? Any thoughts? Okay, let me help us out. Okay, there, Noah came up. Yeah, Noah, you're absolutely right. The voltage-gated calcium channels, right? So remember in chapter 10, the electrical signal came down the neuron and it caused the voltage-gated calcium channel to open so that calcium ions entered the neuron. That discussion we had in chapter 10 highlighted one specific type of voltage-gated channel called a voltage-gated calcium channel. And so there's many others, but what I want to do is tie them to ideas that you've already been exposed to in the course so that you can begin to connect the dots about things you have learned and things I need you to learn going forward. Okay. All right, folks. So there we have the 4 primary types of ion channels you're going to find in the membranes of cells throughout your body. That's figure 1211. Do you have any questions right now? Okay, let's continue. All right, so the next major talking point in this chapter is section 12.4 of your textbook. Section 12.4, and the large heading there is resting membrane potential. Resting membrane potential. And there's figure 12.12 and figure 12.13. Okay, so this is another one of those fairly meaty topics that we need to discuss. And to get it started, I'm going to show you a video this morning. Because I think the video is going to give you a way to mentally imagine these things that are happening with your mind, right? You're going to be able to imagine what these things kind of look like as we have the conversation that we need to document in your notes. So folks, bear with me just a moment. What I'm going to do is show you this video in the upper right. Membrane potentials, excitable cells, and the resting membrane potential. Okay, so that's what I'm going to show you. And just remind you that these videos do exist in Wiley Plus, and encourage you, after you get some rest over Thanksgiving, to review these videos. Okay, so let's get this running here. All right, here we go, folks.
We have the ability to perform intricate, complex movements, make quick decisions, consider creative abstract thought, and communicate freely with others because our neurons and muscle fibers have the unique ability to generate rapid electrical changes in their membrane. These changes are known as membrane potentials. An alteration in the electrical potential surrounding the cell membrane causes a neuron to produce a nerve impulse or triggers muscle fibers to contract. Neurons and muscle fibers are referred to as excitable cells. When the excitable cell is inactive, it is at its resting potential. Changes in the membrane potential may result in either a graded potential, impulses that either excite or inhibit further action, or an action potential, which results in some type of response. In a neuron, it becomes a nerve impulse, and in a muscle fiber, it stimulates contraction. To understand how a membrane potential occurs, we need to first consider the chemicals on either side of the cell membrane. The outside of each cell is bathed in a sodium chloride solution made up of positively charged sodium ions and negatively charged chloride ions. Inside the cell, in contact with the cell membrane, are positively charged potassium ions, negatively charged phosphate ions, and anionic proteins. Separated by the cell membrane, these chemicals accumulate charges on either side, creating a difference in electrical potential. The electrical balance on the membrane is altered when ions selectively pass through the cell membrane to the other side, generating a change in membrane potential. Ion channels, tube-shaped proteins across the membrane, allow specific ions to move through the membrane. The direction of ion flow through a channel depends on the difference in concentration of electrical charge action on the ion. Ions passively diffuse through a channel from higher to lower concentration. They are also attracted by opposite charges or repelled by like charges from other charged molecules. Thus, the direction of ion flow depends on the balance between both chemical and electrical forces acting on the ion at any given time. Typically, sodium ions tend to enter the cell through an open sodium channel, and potassium ions tend to exit the cell via potassium channels. There are 4 types of channels through which ions can pass. Leak channels are typically always open. The 3 types of gated channels, chemically, voltage, or mechanically gated, must open their gate to allow the passage of ions. A leak channel allows only a trickle of sodium or potassium ions to diffuse through the membrane. Leak channels contribute greatly to the generation and maintenance of the resting membrane potential. A chemically gated channel, also referred to as a ligand-gated channel, has a receptor associated with it. When its receptor is occupied by a chemical known as a neurotransmitter, the gate on the channel opens, allowing a specific type of ion to diffuse through. The gate will remain open as long as the receptor is occupied. A voltage-gated channel opens at a specific voltage, referred to as the threshold. For example, the threshold for a voltage-gated sodium channel is around -55 millivolts. When the membrane voltage increases from its resting state of about -70 millivolts to the threshold, the channel gate opens and sodium diffuses through the channel to the other side of the membrane. Some voltage-gated channels open quickly, while others are slow to open. Even though they may reach threshold at the same time, the speed at which they open causes significant changes in membrane potential. Some voltage-gated channels have an additional gate referred to as an inactivation gate, which temporarily plugs the channel so ions cannot pass through, even if their activation gate is open. Its purpose is to ensure that ions do not pass through the gate at an inappropriate time. Mechanically gated channels open when pressure, touch, or stretch is applied to the membrane. They are found in sensory receptors, located primarily in the skin, to inform the nervous system of mechanical changes in the surface of the body resulting from conditions in its environment. An ion pump is a type of membrane transport protein. It requires energy to push ions from an area of lower concentration into an area of higher concentration. The ion pump most important in the maintenance of membrane potential is the sodium-potassium pump. Using ATP as its energy source, it is able to pump 3 sodium ions out of the cell and 2 potassium ions into the cell at the same time. Leak channels and ion pumps complement each other to maintain a constant electrical potential and level of ion concentration. The resting membrane potential voltage is determined by the concentration of electrically charged ions on either side of the membrane. It is established primarily by leak channels. There are more potassium leak channels than sodium leak channels, so more positively charged potassium ions pass out of the cell than positively charged sodium ions diffuse in. Also, sodium-potassium pumps move more sodium ions out of the cell than potassium ions in. Leak channels and ion pumps complement each other to maintain a constant electrical potential and level of ion concentration. As positively charged ions move out of the cell, they accumulate on the outside of the membrane, giving it a positive charge. The inside of the membrane is electrically negative because the negatively charged phosphate ions and anionic proteins lack channels, so they remain in the cell, and also because more positively charged ions move out rather than in. This establishes an electrical difference or potential on the membrane. Since the inactive cell membrane is negatively charged inside and positively charged outside, it is considered to be polarized. The voltage created by this distribution of ions is known as its resting membrane potential and is maintained everywhere on the cell membrane. The excitable cell remains in this state until it receives a stimulus.
Okay, folks, so there you have it, all of life in about 7 minutes, right? So again, I want to encourage you to review the 2 videos, one I showed last lecture and this one this morning during a private study time. Are there any questions about these ion channels or about the video that you would like to ask? Okay. All right. So we're going to continue to push forward. Okay. So please let me know if you have questions. All right. So let's take some of these ideas that were presented about the resting membrane potential in the video, and again, which are presented basically in figures 12.12 and 12.13 of your textbook, and document them in your notes. Okay. So first, when we talk about a resting membrane potential, we're talking about a neuron or muscle cell that's at rest. And what we mean by at rest is that the cell is not propagating an action potential. There's no electrical signal spreading along the length of the axon. Okay. So to establish the resting membrane potential, we have to appreciate the electrical status of the interior of the cell compared to the exterior of the cell in the extracellular fluid. So let's state it this way. On average, the cytoplasm inside the cell and the extracellular fluid outside the cell have an even mixture of positive and negative charges, where those charges are associated with a variety of ions and molecules that have a charge. Okay, so the take-home message is that the average electrical status of the cytoplasm and the average electrical status of the extracellular fluid, again, on average, is neutral. Okay. Okay, so let's just kind of draw this out a little bit in our document. I mean, the figures in your textbook do a fine job as well, but sometimes it just helps to redraw, redraw what you know, because it helps solidify your understanding of things. So let's just take a second to do that. What I'm saying here with statement number 1 is that if we have the plasma membrane of a cell here, and we have the inside of the cell right here, which is obviously the cytoplasm, and we have the outside of the cell here, which is the extracellular fluid. And we would note the following situations, okay? In the interior of the cell, in the cytoplasm, we pretty much have an even number of positive and negative ions. So that means, just as a rough example, I could draw 4 positive ions here. And to balance that out so that I have neutrality, I would need roughly 4 negative ions, right? So this here is a neutral situation. The same thing is going to be true outside the cell. Sorry about that. My colors are not working here. Okay, so this is also an approximately neutral environment. That is true. Now, the membrane itself is a little different. The membrane itself is a little different. So when we talk about the charge across the membrane, I'm going to be talking about this membrane here and the fact that there is an inner and outer surface to the membrane. While it is true that the interior of the cell, for the most part, is neutral, and the exterior of the cell, for the most part, is neutral, it is also true that the membrane is not neutral. Okay, so for example, on average for a resting cell, typically the inside of the membrane has an abundance of negative charges. So let's say for every 4 negative charges, I might have 1 positive charge, just as a rough idea here of how this would look. Okay, so what I'm telling you is that the inner surface of the plasma membrane in a resting cell is overall negative. Okay. Overall negative. And on the outer surface of the membrane, I have the exact opposite situation where I would have roughly 4 positive charges for 1 negative charge. So that the outer surface of the membrane, on average, is overall positive. Okay, so the interior of the cell is negative. The exterior of the cell, the interior of the cell is neutral. The exterior of the cell is neutral. But if we focus our attention specifically on the membrane, the plasma membrane, the inner surface is negative and the outer surface of the membrane is positive. Okay. All right, so let's take the ideas that were shared here in the drawing and put some additional words to things. Do you have questions right now that you'd like to ask? Okay, question. Okay, so let's continue here to document these ideas. Okay, so I'm just going to document in words kind of what I'm trying to suggest in the drawing. Okay, along the inner surface of the plasma membrane, negative ions tend to accumulate. And on the exterior surface of the plasma membrane, positive ions tend to accumulate. Okay, and so you might ask yourself, well, why is this the case? Why is it that the inner surface of the membrane has an overall negative charge and the exterior surface of the plasma membrane has an overall positive charge? What explains that? Okay, so let's address that. Why? Okay, the answer to why, or perhaps how, comes back to these leak channels that I started our conversation with. Okay, remember, there are more potassium ion channels in the membrane than there are sodium ion channels. And so potassium ion channels are designed to allow potassium ions inside the cytoplasm to escape onto the exterior surface. So since we have more potassium ion channels than sodium ion channels, we are leaking more we are leaking more potassium ions. We are leaking more potassium ions out of the cell. Potassium ions have a positive charge, so therefore these positively charged potassium ions leak out of the cell and accumulate on the exterior surface. That explains why there's an overall abundance of positive charge outside. Because those potassium ions are leaving the cytoplasm, whatever negative charge is inside the cell is not going to be masked anymore by the potassium's positive charge, which means the inner membrane takes on an overall negative characteristic. Okay, so that's a lot of words here. Let's try to be succinct here. So why does the membrane have the charge it has? Okay, the behavior of the leak channels explains this. Because there are more potassium ion leak channels that allow potassium ions to exit the cell, positive charge accumulates on the exterior surface of the plasma membrane. As the positively charged potassium ions leave the cytoplasm, the inner surface becomes negative due to the absence of the positive charge. Okay. All right, folks. So, question in the chat window. Is this making sense? Is this making sense?
I have a question.
Okay.
So is potassium, the positive potassium ion, like free flowing?
No, what do you mean by free flowing?
Like it's not like attached to any other ion to create like a molecule?
Okay, yes, thank you. Yeah, you are correct. That is true. The potassium ions are individual atoms with an electric charge that are not associated with another ion. So in other words, it's not a salt. It's not potassium chloride. Like if I had positive potassium and negative chloride and they joined together, they would form a salt, potassium chloride instead of sodium chloride. That's not what's happening here. The potassiums are free floating, and you might say, well, why is it free floating? Because while there are some negatively charged chloride ions around, there's so much water in the cell that the water kind of separates those ions for the majority of their lifetime. So you do have this group, this pool, this civilization, this community of individual potassium ions that are not bound to anything at the moment. And in that state of existence, those individual potassium ions do free float through the leak channel proteins to get to the other side, the outside of the cell. Does that help, Noah?
Yeah, it was my main question was why wasn't it like binding itself to the chlorine or any negatively charged ions in there? But I guess you said that there's water in there that's keeping it from doing that.
Absolutely. Absolutely. It's a very good question that you ask, and it's a very reasonable question. You could expect, Noah, that there is a population of potassium chloride in the cell. There are going to be some potassium ions that are actively bound to chlorine, but there's not enough chlorine around to bind up all the potassium. And so for that reason, you're going to have a subset of potassium ions that are not actively attached to anything else and have the luxury of being a quote unquote bachelor so that they can go through that channel. Okay. All right. Good question. And please ask. And also don't forget, I made the comment about there being water present. One of the things about understanding how cells work is that when we explain the movement of ions or molecules, like sodium ions or things like this, we typically ignore the fact that all of this movement is happening through a swimming pool worth of water. Water influences the chemistry inside a cell, and including the electrical state of molecules and ions that might or might not be bound to each other. Okay. So let's continue here. All right, so next, number 3, the resting membrane potential of a typical neuron is minus 70 millivolts. Okay, minus 70 millivolts. You see that in figure 12.12. Okay. The resting membrane potential of a typical neuron is minus 70 millivolts. Well, what are the factors that help maintain that minus 70 millivolt membrane potential? What is contributing to that? There's basically 3 things, and this gets to actually figure 12.13. Okay. All right, so first point, there's going to be an unequal distribution of ions in the extracellular fluid and the cytosol, an unequal distribution of ions, okay? So your book is going to do a better job of this than I could potentially draw. And I'm gonna reference this to everybody, but in particular to you, Noah. Noah, do you notice in figure 12.13 of your book that on the outside of the cell, in the extracellular fluid, your book is drawing quite a few negatively charged chloride ions. But if you compare that to the inside of the cell, the cytosol, there's relatively few chloride ions. And so this kind of gets to your issue of, well, why doesn't the chloride ion inside the cell bind? Why doesn't the potassium ion inside the cell bind to the chloride ion? It's because the cell maintains this gradient where most of the chloride ions are sitting outside the cell rather than inside the cell. So they're just not in the right location to bind the potassium inside. So potassium remains a bachelor to move through the leak channels and get outside. Okay. All right, so the unequal distribution of ions in the extracellular fluid and cytosol. Okay. The extracellular fluid, the ECF, if you will, I'm going to abbreviate it that way, ECF, just for note taking. The extracellular fluid is abundant in sodium ions and chloride ions. And the inside of the cell, the cytoplasm, is abundant in potassium ions and negatively charged anions, of which one very common type of negatively charged anion would be phosphate groups, the PO4-3 things that you see drawn. PO4-3 groups. Okay. Second factor that influences the resting membrane potential is the inability of these anions to actually leave the cell. These phosphate anions, these PO4 groups that have a -3 charge, well, there's 5 atoms there, right? 1 phosphate, 4 oxygen, PO4. So that forms a group or cluster of atoms that are simply too large to float through the plasma membrane. So these negative ions, these negative groups of atoms, they stay inside the cytoplasm for the most part. Okay, the inability of most anions to leave the cell. Phosphate anions are too large to diffuse through the plasma membrane. Okay, and then the third and final point is this big fancy word called electrogenic, the electrogenic nature of the sodium ion, potassium ion ATPases. Okay. A lot of times people call these sodium potassium ATPases the sodium potassium pump, right? The sodium potassium pump. Okay, so let me talk a little bit about that. That word electrogenic is simply a fancy way of saying that these sodium ion, potassium ion pumps have the ability to change the membrane potential. That's what electrogenic means, the ability to change the membrane potential. Okay, and so you saw from the video that these pumps actually move ions from an area where there is a low amount of ion to a high amount of ion. So they're going against the concentration gradient. And you may recall from our time talking about transport processes in chapter 3 that when you move against the concentration gradient, that's going to require the cell to burn energy. That's why the sodium potassium ATPases, that you see in figure 1213 are breaking down a molecule of ATP to power the pumping action to push the ions against the gradient from low to high rather than high to low. Okay, and so we should say this here. Okay, remember that leak channels, I'll say it this way. Remember that sodium ion leak channels tend to leak sodium ions into the cell. And potassium ion leak channels move potassium ions out of the cell. Okay, that's how the leak channels move these ions. So the job of the sodium potassium pumps is going to be to move the respective ions in the opposite direction of the leak channels. So that means the sodium potassium pump is going to pump sodium ions out of the cell, and the sodium potassium pump is going to pump potassium ions back into the cell, and it's going to burn energy to accomplish that goal. Okay. Sodium ion pumps move ions oppositely to the leak channels. Okay, so that means that sodium ion pumps pump sodium out of the cell, and they pump potassium into the cell. And not only that, but these pumps require ATP, so energy, to do their work. So it might be an odd idea to consider this, but even maintaining a resting membrane potential during sleep, even maintaining a resting membrane potential during sleep when you're not conscious in any way, still requires the expenditure of quite a lot of biochemical energy. It still requires the expenditure of a lot of ATP fuel simply to maintain the proper membrane potential, so that if part of your brain wants to dream and create this dream state, this dream world for you, that it can do so because the membrane potentials are being maintained at their proper charge. Okay. All right, folks. Any questions right now? This is tough stuff. And unfortunately, it's going to require a considerable amount of effort on your part to really, watch videos, review the notes that I'm giving you, maybe review the recording that we're having right now, read the textbook, take your own notes, do practice questions before it all starts to come together and you start to say, well, okay, I think I understand this now. All right, so with the resting membrane potential established, and we've kind of tried my best here to describe what it is, how it is maintained. With the resting membrane potential established, we now need to talk about, well, okay, what happens when you do receive a stimulus? When you detect an odor from a skunk sitting outside your room, what happens? Well, basically what happens is that the resting membrane potential is altered by the stimulus. The resting membrane potential changes from -70 to something else. Typically, that something else is going to be something that makes the membrane potential a little bit more positive. And if it reaches a critical threshold known as threshold potential, which is for most neurons -55 millivolts, then you will generate and action potential, and all kinds of sophisticated things start to happen, right? Such as, oh, there's a skunk sitting out my window. I'm consciously aware of it. I don't want to smell that. So I'm going to tell my skeletal muscles to contract so I can walk away from the window so I don't have to be exposed to that horrible odor anymore. Okay. All right. So here's a big question. What happens when your body experiences some type of stimulus? Okay. The quick answer is the resting membrane potential is disturbed. Okay. And so this is going to happen in two stages in terms of our abilities to be aware of what's happening to our bodies. What sound am I hearing in my ears? What sight am I seeing in front of me? As you watch the Macy's Thanksgiving Day Parade Thursday morning, what are you seeing with your eyes? What are you hearing with your ears? If somebody's cooking a turkey in the oven, what are you smelling in the air? Okay, the resting membrane potential of your sensory apparatus in the eyes, ears, nose, surface of the skin when you get a hug. Those sensory apparatuses, the neurons in those pieces of anatomy are going to have their resting membrane potentials disturbed by the stimulus. Okay. All right, so we need to break this down into two stages. First, we need to talk about graded potentials. All right, graded potentials, which we defined 1 or 2 class periods ago, I can't quite remember. These, remember, are the type of electrical signal in the nervous system that are used for very short distance communication. Okay, so as an example, if you were in a city and you were walking along the street and you took your hand and you rubbed your fingers up against the concrete wall of a big building in the city, as you walked past the wall and your fingers kind of bounced off the cement of the wall, you would get a sensation, because the stimulus is that your fingers are bumping up against the cement repeatedly as you walk across the building. That's going to create graded potentials in the nerve fibers embedded in the dermal layer of your skin. Okay, so number one, most graded potentials occur in the dendrites and cell bodies, in the dendrites and cell body of a neuron. And of course, there might be multiple neurons that are activating when you rub your fingers across some building. But most graded potentials occur in the dendrites and cell body of a neuron in response to some kind of sensory stimulus, physically touching an object, smelling an aroma, a fragrance, a perfume in the air, photons of light hitting the retina in the back of the eyeball, sound waves striking your eardrum and vibrating it inside the inner ear. All of this is going to generate graded potentials in the neurons that are in that region of your body's anatomy. Okay. Most graded potentials occur in the dendrites and cell body of a neuron as the neuron is stimulated. Okay. Number two, this typically occurs when you receive a stimulus that causes a mechanically gated channel to open, right? We just defined mechanically gated channels. So think about it. Sorry about the noise. I got an ambulance outside going past here. mechanically gated channels are going to open when you brush your fingers against the wall of the building that you're walking past in the city. That's one possible stimulation. You could also have ligand gated channels open up, right? When the aroma of the turkey cooking in the oven reaches your olfactory receptors in your nose, those neurons are going to have the molecules in the air that represent the odor, the fragrance, the sense of the cooking turkey. They're going to bind to receptors embedded in your nose, and those channels are going to open, and ions are going to flow across the membrane. So this typically occurs when a stimulus causes a mechanically gated channel, and I'll put it here, rubbing your fingers against the wall. Or ligand-gated channels, such as the odor of a cooking turkey, causes these channels to open or close. Okay, so the graded potentials typically occur when a stimulus causes a mechanically gated channel or a ligand-gated channel to open or close. Okay. Now there's two ways that this can actually happen. Okay. The most common way that we are currently talking about is, rubbing your fingers against the wall or having molecules in the air from the turkey bind to receptors in your nose. In that situation, the stimulus is causing the inside surface of the plasma membrane of the neuron to become more positively charged. When that happens, the neuron is experiencing a depolarization event, which is the same thing as an excitatory signal. Okay. Rubbing your fingers against a wall would cause the neurons in your fingers to depolarize. This means that the inside surface of the plasma membrane becomes more positively charged. You ask yourself, well, why did the inside surface become positively charged? Well, because the mechanically gated channels opened, allowing the sodium ions into the cell. So we're bringing positive charge into the cell. Okay. So there's a fancy word here that you're going to want to spend some time thinking about, and that is depolarize or depolarization. Okay. Oop. And let me just put this in your notes here. Okay. Depolarization is basically equivalent to an excitatory event. I mean, you're going to feel something. That's what I mean by excite. You're going to feel it. You're going to be consciously aware of it. Okay. There is another possibility, however, that can occur. And for this one, we haven't really talked about it yet, and I need to get a little bit deeper into the chapter before it's going to make a lot of sense. But imagine this scenario. Okay, let me ask you a question in the chat window. do you imagine that you want excitatory events in your nervous system as you are trying to fall asleep? Do you want your nervous system to become agitated while you're trying to fall asleep? Not really. So there's another aspect to graded potentials. Graded potentials can be involved in processes that try to calm your nervous system down. And in that case, we're typically talking about neurotransmitters being released in the brain by certain neurons that are trying to communicate to other neurons to, hey, calm down. Calm down because I need to fall asleep, okay? So in such a case, we have the opposite of depolarization happening. When you try to fall asleep, requiring you to calm down, then some neurons will hyperpolarize. They will hyperpolarize, and hyperpolarize means that the inside surface of the membrane becomes even more negatively charged than it was at rest. Okay, so the fancy word here you're going to want to focus on a little bit is hyperpolarize. Okay, hyperpolarization. Hyperpolarization basically amounts to an inhibitory event. Okay, an inhibitory event such as we're trying to get the electrical activity in the brain to calm itself down so I can actually fall asleep. Okay, so these are the two different types of graded potentials that you might encounter depending on the stimulus. While you're enjoying Thanksgiving dinner, the odors and flavors, they're going to generate depolarization events in the sensory machinery of your body, which is going to excite your nervous system, and you're going to like, oh, that's delicious turkey, just wonderful. But later on, after you've gorged yourself, right, you're going to stumble into the living room and sit down on the couch and probably want to take a nap. And so there's going to be neurons in the brain that are actually releasing neurotransmitters to communicate with other neurons that normally are active to keep you awake. And so these neurotransmitters are trying to inhibit the activity of the waking neurons so that you actually fall asleep so your digestive system can do its job. Okay, that's what the graded potentials would do. Okay, let's continue here. Any questions? Okay, so graded potentials are things that are described in section 12.5 of your textbook here. Section 12.5, right? And you do have figure 12.14 there, and you have figure 12.15 there. These are things that you're going to want to look at during your private study time. You know, if I were to cover literally everything like your textbook does, I would need a month worth of lecture, and I'm already running out of time here. So I want to leave it to your capable efforts to do the study you need to do. But I'm going to make a couple other comments here. Okay, we're talking about graded potentials. Okay, graded potential is the first type of electrical signal that will take place when you receive a stimulus. It's the first type. Okay, the term graded means that the electrical signal varies in amplitude, and that's what figure 12.16 is trying to show you. The term graded means that the electrical signals vary in amplitude, okay? And so I'm just going to ask a question. I'm hoping you're able to see figure 1216. So I'm going to ask a question about figure 1216. Here we go. In figure 1216, which hump, green, blue, or purple, has the largest amplitude? Which hump has the largest amplitude? Okay, thank you, Shay. Shay's absolutely right, and I'm going to run with that because I'm running out of time here. So my point here is that if you've identified the purple hump, then you do have the basic appreciation of what amplitude is, right? Amplitude is the distance between the top of the hump and the bottom of the hump. The distance, the vertical distance between the top of the hump and basically the x-axis in that graph, okay? So the term graded means that the electrical signals vary in amplitude, and the amplitude itself is controlled by, which is controlled by two things. First off, the number of ion channels that have opened or closed, and the length of time each ion channel has stayed open or closed. Okay. Okay. So one last thing, and then I think we'll wrap it up for this morning. The last thing is this idea of summation, because graded potentials work over very short distances. They kind of activate, they spread a little bit of distance along the anatomy of a dendrite or potentially a cell body, and then oftentimes they just stop because they're only capable of moving a small distance because of the way the anatomy of the plasma membrane has been designed by Mother Nature. So if a graded potential moves a short distance and does not reach the axon hillock of the axon, there's a very good chance that you will never generate an action potential from the graded potential. And if you don't generate an action potential from the graded potential, then you're not going to get electrical signal that goes up into your brain, and you will not be consciously aware of the stimulus. So what I'm talking about, imagine the most gentle breeze outside when you're outside, the most gentle breeze that is very delicately, just very, very slightly moving the hair fibers on your arm, but it's doing so in such a way that you pretty much aren't paying any attention to it. You're kind of ignoring it because it's not a violent wind. It's very, very, very gentle. So your hair follicles are just bending a little bit, but it's not bending enough to generate enough graded potentials to reach the trigger zone of the axon hillock. So you're not consciously aware that the hair follicles on your arm are actually moving a little bit, okay? I mean, most times, we pay attention to those things that our brain is aware of, and our brain can only be aware of things when there's been an action potential generated. The action potential can only be generated from graded potentials. But a lot of times graded potentials individually are not strong enough to generate an action potential. So Mother Nature came up with a solution, and that solution is called summation. Okay. Because graded potentials, and I promise I'll stop after I get this documented, because graded potentials act over very short distances, they tend to die out quickly without generating a subsequent action potential. Okay, if you don't generate the action potential, then your brain's not going to be aware of the stimulus, not going to be aware. Okay, so the impact, the strength and duration of a stimulus can be influenced by the fact that you generate multiple graded potentials that come together. They kind of overlap each other in this summing mechanism, the summing phenomenon, which is being illustrated in figure 1217. If you look at figure 1217, you see stimulus 1 and stimulus 2. So here's the idea. Let's go back with the idea of walking along a building in the city, right? Stimulus 1 would be the first moment your finger vibrates a little bit when you touch the side of the wall of the building. And then as you take your next step and physically move your body forward, your finger bounces off the position it was and then hits the next position. As you hit the next position, that's stimulus 2. So as you're moving along, you're vibrate, 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 vibrate. Each vibration is stimulus 1, stimulus 2, stimulus 3, stimulus 4. So as you continue that walking over a duration of time, the graded potentials are triggered multiple times and they start to build on top of each other. That is summation. If that summation is effective, then you get an overall summing amplitude there, which is much higher than the amplitude of each individual stimulus event. And that summing amplitude is strong enough to reach the axon hillock of the neuron. And when you do that, bang, you can trigger the action potential. Okay. Both the strength and duration of a stimulus can cause multiple graded potentials to form and overlap with each other. This is a phenomenon called summation. And it is true that summation signals are usually strong enough to reach the axon hillock of an axon and trigger the creation of an action potential, which will ultimately make your central nervous system aware of the stimulus. Okay, let me read those notes and we'll wrap up. The summation, both the strength and duration of a stimulus can cause multiple graded potentials to form and overlap with each other. This is a phenomenon called summation. That is figure 1217. The summation signals are usually strong enough to reach the axon hillock of the axon. Okay, let me get that fixed. And trigger the creation of an action potential, which will ultimately make your central nervous system, and therefore your conscious awareness of the stimulus, possible. You are aware when Aunt Nancy gives you a bear hug Thursday morning. Okay, it's going to start off as localized graded potentials that summate to an action potential triggered at the axon hillock of the nerve, of the axon as part of the nerve, that travels up to the brain and say, okay, I'm getting a bear hug from Aunt Nancy. Okay, folks, I think that's about as good as I can do here. We haven't finished this chapter, so what I will have to do after Thanksgiving is continue in this chapter. I'm hoping maybe 30 minutes more of time, then I'll be done with it. Then we got to start the next chapter. And so I expect that I might have to use a little bit of lab time for lecture purposes to get us up to speed so you are at where you need to be at for your upcoming final exam. Okay, any questions, folks?
Yes, I have a question. I actually have two. The first question is, I would like to review the recordings for this chapter. I don't know where to find them.
Okay, okay. Shay, all the recordings are being hyperlinked in the lecture portion of Wiley Plus. So do you know how to get to the lecture portion of Wiley Plus?
Yes, I'm there now.
Okay. Within Wiley Plus, along the left-hand side, you should see a button that says Pages. Do you see Pages?
Yes.
Okay, go ahead and open that up. And then once you're in Pages, do you see a line that says links to lecture recordings?
No.
You don't. Okay, hold on a second. You should see it. Let me share my screen with you and show you what I'm talking about. Okay. All right. Okay. So first off, and I would emphasize, remember, Shay, that you have a lecture portion to Wiley Plus and you have a lab portion to Wiley Plus. So make sure you're in the lecture portion. If you're in the lecture portion and you go down to Pages, which I'm doing.
Found
right.
it. I found it.
Okay, all right. So links to lecture recordings, you should see every lecture of the semester there, and it's hyperlinked. When you press that link, it's going to take you to a recorded video that's stored on YouTube.
Okay.
Okay.
My second question was about, thank you for that. My second question is about the lab. So in lab last week, you said that for the spinal cord cross section, we needed to use figure 13.1 because visible body didn't have that section. So I just wanted to confirm some terminology
Okay.
that looks different.
Okay. We can do that. Let me just pause what I'm doing here, and I can definitely do that with you.